Welcome everybody. We are live from California for today's webinar with Inscripta. Inscripta is based out of Boulder, Colorado, and has other offices here in the Bay Area and in San Diego. And we're going to be joined today by Andrew Garst, who is the principal scientist and founder of Inscripta. We're just going to wait a few minutes for people to continue joining before I bring in Andrew to talk about precision editing for genome scale evolution. That's the topic, and we're going to be looking underneath the hood for something that many of us have been waiting for, because Inscripta has been around now for a few years and is about to make a very big splash at SynBioBeta in October. Kevin Ness is going to be giving the welcome keynote, and he's also the presenting sponsor of SynBioBeta 2019, which is going to be at SVN West in San Francisco, October 1 through 3. Many of you have already bought your tickets to come join us, and the most important thing that the presenting sponsor gets to do is throw the big welcome launch party at the end of day one, which is on Tuesday, October 1st. So I'm very excited to be working with Inscripta. I'm John Cumbers, and I'm the CEO and the founder of SynBioBeta, where tech meets bio and bio meets tech. And our webinar series features conversations with synthetic biology's leading thinkers and doers on building a better world with biology. Now, today is going to be an interactive webinar, and if you look at the bottom, you will see a Q&A box, and you can type in your questions into that box, and Andrew or I will be able to answer them in real time. We're going to talk about the challenges of genome engineering today. We're going to be looking at the tools and technologies that Inscripta is using to overcome them. And we're going to be talking about Andrew's vision for digital genome engineering in the years to come. And we are recording this webinar, so you'll be able to watch it later on. And we also welcome any feedback on the format. You can either send it to us via email, or again, you can put it in the Q&A in the comments section. So with that, I'm going to dive in and welcome Andrew. Andrew's interests reside in the study of RNA biology and the development of RNA-based tools that can be leveraged for applications in biotechnology and medicine. He's particularly interested in building forward engineering solutions that enable rapid design and characterization of living systems for basic and translational research by combining user-friendly software, automation, and robust biological performance. He enjoys working in cross-disciplinary teams that value creativity and innovation and favor empirical problem-solving approaches over expert dogmatism. Andrew has a PhD in biochemistry from CU Boulder, and he is the principal scientist at Inscripta, who's developed all of the core chemistry that you're going to hear about in this pretty amazing piece of machinery. Welcome, Andrew.
Thanks, John.
Pleasure to have you here.
Yeah, thanks for having me. This is great.
So we're going to dive into a presentation that you've got for us shortly, but I wanted just to ask a couple of background questions. Can you tell me a little bit about how you came to co-found Inscripta?
Yeah, so I was working as a postdoc at CU, and the stated goal of my research there was to try to develop genome-scaled editing tools, explicitly with the goal of being able to track edits at population scale. So when I joined the Gill lab, they were pretty immersed. This was Ryan Gill at CU Boulder. They were already immersed in the genome editing world. So they had developed a couple genome-wide technologies that were non-nuclease based, and my research goals were to bring on the nuclease-based engineering systems in that lab. Sorry, we're having video issues trying to turn off the video. So I spent the first year bringing on CRISPR into Ryan's lab, and then we spent another couple years really working on trying to scale CRISPR to do the kinds of genome-wide applications that we were interested in for the directed evolution side of things, because when we inherited CRISPR, it was really a one site at a time technology. That work translated into what I'll talk about today as the CREATE technology, which we published in Nature Biotech. And yeah, from there, Ryan and our other co-founder, Tanya, they did a good job of pitching this to investment teams that we eventually raised funding and started the whole Inscripta mission to really democratize all of this for the broader research community. So.
And what you're going to be launching at the conference in October is a one-stop shop piece of hardware that's going to sit on the lab bench of many labs around the world. And the big deal is the scale of what you can do with this machine that you can't do elsewhere. Is that right?
That's right. So yeah, rather than a large foundry setup where you'd have lots of automation and moving a lot of moving parts to move sample wells around, what we're building is sort of a one-stop shop, as you said, where everything's self-contained on a single instrument, and you can deliver tens of thousands to even hundreds of thousands of edited cell libraries at a time. So that's really the scale and scope of what we're trying to provide for our customers. Got it.
And CRISPR can theoretically let us remix entire genomes, but instead of true forward cell engineering, we still seem to be tinkering with one gene here and one gene there. What's been holding us back so far?
Well, I think that when we started in CRISPR, there was some dogmatism about the workflows that you needed to do. There was a lot of prior art on using single-stranded oligos as the donors, and I think people really bought into that's how you had to do genome editing to get high efficiency. So there was, I think, the efficiency debate dominated for some time, and we sort of reimagined the way in which you could put all the parts together and deliver both the donor and guide pieces to be able to get a scalable system, because we were really stuck at we could do this for a single locus. We could do many edits in a single locus, but we really couldn't scale it beyond that until we adopted the CREATE approach.
Got it. So we're going to go into the CREATE biochemistry and the Nature Biotech paper that you published on it. Help us understand, for everybody listening, what the difference is between forward engineering and reverse engineering.
Yeah, so we see forward engineering as a pre-programmed set of questions that you can state to your biological system. So there's design inquiry that goes into the upfront part, and that's really sort of a massively parallel hypothesis testing machine, versus reverse engineering, where you're sort of seeing interesting traits or phenotypes pop out of some traditionally like adaptive laboratory evolution or other diversity generation process, and then backing out which mutations you made. We're providing a way to sort of close that cycle and do it in a forward engineering process.
Great, excellent. So we're going to dive into your presentation shortly. Final question is thinking about the microbial hosts that the platform is compatible with. Is it just E. coli, or what's the range of organisms that you can put into this machine?
So yeah, at first customer ship, it will be E. coli and Saccharomyces cerevisiae. There are plans to expand that. So both in the number of different strains, there are different strains of E. coli and cerevisiae. But then beyond those two, there are plans to develop a mammalian platform, mammalian cell platform. It'll be select lines that we're going to provide that for, and that I think we're looking at 2020 for launch on that.
Got it. Okay, fantastic. So I just want to do a technical check to make sure that we're recording this, because it says recording paused on my screen. So just want to check that it's working before we dive into the slides. It's looking like everything's good. So Andrew, if you want to bring up the slides, we will dive right in.
Alrighty.
And while Andrew's doing that, I just want to remind people this is live. So if you have questions during the talk, no matter of their nature, if it's a dumb question, we like those. If it's a technical question, we like those too. So just post them in the box, and we'll take them as they come in. Okay, so over to you, Andrew.
All right, great. Yeah, so to tell the Inscripta story, I thought it would actually be good to kind of put some context around where we entered the larger directed evolution tools scheme. So I'm going to break this talk into 3 sections where I first introduce CRISPR and the directed evolution tool space. Then I'm going to talk about the create technology that we launched Inscripta with. And finally, I'm going to talk about Inscripta's goals of democratizing genome engineering for the broader community. So for the introduction to CRISPR, what I like to do here is break it into both the non-nuclease and nuclease-based technology. So on the top, you have the non-nuclease technologies and some of the achievements people have reached using non-nuclease-based engineering. So this sort of engineering spans all the way back to the advent of agriculture. People were employing, at the time they didn't know the mechanism, but they were employing recombination to be able to do selective breeding and trait selection for different animals and crops. It wasn't until 1859 that Charles Darwin started to put some framework on what the selective units were and help us understand the mutability of organisms. And then by 1914, people actually started to do some of their first experiments in a laboratory setting where they employed chemical mutagenesis. And we also started to inherit our first concepts of a fitness landscape from C. Wall Wright in 1932. So that was all before we even really took directed evolution into the lab. And it wasn't until the 1960s that we really started to see laboratory examples of directed evolution, starting with some of the RNA evolution experiments that Saul Spiegelman demonstrated, where an RNA could self-evolve for replicative purposes. You then saw through the advances in oligosynthesis, Smith showed that you could do site-directed mutagenesis. So oligo-mediated mutagenesis started to come online there. And fast forward to 1980, we started to understand how to deliver DNA into the nucleus. This came out of Mario Capecchi's work, which he received the Nobel Prize for the nuclear delivery of DNA, showing how transgene expression could work. 1982, we started to understand how to do some directed evolution using phage display. So this is starting to, look more like what we think about in today's world of high throughput directed evolution, where you can create massive library of protein variants, display them on a phage, and then select for the best binders and activities and run that in a recursive mode. By 1987, site-directed mutagenesis was shown to be first demonstrations that you could do that in mouse embryos, again from Mario Capecchi, really started to open people's minds to, hey, like we can directly impose, new code into living systems and test the phenotypic outcomes. And then in the 1990s, we saw the generation of a number of different new tools to speed up this evolutionary process. So starting in 1993, Francis Arnold started to employ an error-prone PCR process that could speed up the mutation rates as you amplify different genes for diversity generation. And she, as you see down there at the end, 2018 on the right side here, let's see if we can you see the mouse? She later she just recently won the Nobel Prize for the accumulation of that work among the many others listed here. Pim Stemmer also brought online DNA shuffling, where again, this process is actually like what was used in the selective breeding back here in the ancient times, except for that now it was done in the lab at a very high rate using enzymatic principles to speed up the rate of recombination between beneficial variants. So he showed this first at the single protein and later at the pathway and genome scales over the course of about a decade. You also started to see microarrays make their way into the genome editing world. So people started to understand how to use barcoding principles to make gene knockouts, genome-wide knockout libraries in yeast out of Ron Davis's lab at Stanford. And you also started to see in the early 2000s structure-guided approaches where people were, basically able to predict where you'd have the biggest impacts on a protein function and guide the directed evolution search because you have such a large search base with which to operate in for any directed evolution problem. 2003, you started to see some machine learning meets directed evolution with things like the ProSAR algorithm. This was published by Richard Fox, one of my esteemed colleagues here at Inscripta, when he was at Maxigen, and that really started to hyper accelerate some of the things people could do in the industrial setting in terms of beneficial traits being selected on rapid timescales. This sort of work led to some of the directed evolution for enzymes like a transaminase that could make stigoliptin that came out of Codexis. This is a multi-billion-dollar-a-year drug that was developed using directed evolution principles. So you started to see directed evolution having an impact in the marketplace directly from the sped up lab processes. There's also tools like multiplex automated genome engineering out of the Church Lab that started to get people thinking more on how do we hyper accelerate this in rational ways at the genome level. And then of course, there's some recent work that's been done out of both Jeff Buca's lab and at SGI with Dan Gibson, where whole genomes have been assembled from the starting point of synthetic oligos up to the whole genome scale. So a lot of interesting things were happening in the non-nuclease world and overlapping with what was happening in the nuclease world. But I like to show this because some of what these guys were employing, like in studies here in the 2006 stigoliptin story, were very high throughput in their ability to generate diversity. Meanwhile, the nuclease editing technologies were still lagging way behind. So with the nuclease-based technologies, the story is a little different. If we shift down to the bottom here, you had 1972, Paul Berg discovered restriction enzymes, and people started to use those for cloning purposes. But it was as early as 1988 that people realized that with the homing endonuclease, the Ho nuclease, you could create high levels of recombination through a double-strand break within the chromosome. So by the 1980s, we already started to have some idea that, hey, if we can figure out how to do this better and at more sites, we have a tool here to modify genomes directly. In 1992, people also started to see that with another meganuclease, the SCI1 nuclease. These studies were both done in yeast. And then it was 1993 where we first started to get hints that there was something interesting in these CRISPR loci, and they were being found in multiple different organisms across samples collected out of the ocean. 1996, you saw the first FOC1 ZFN programmable nuclease demonstration. So this was really starting to resemble what we're doing in the modern. Is my mouse moving there? Sorry. This is really starting to resemble what we're doing now, where we want programmability and predictability in the cut of where we're going to precisely edit a genome. Then in 2002, the first in vivo demonstration of that technology was shown again by the Chandra Segaran lab. 2008 is actually, many people don't know, this is where we start to get into the more CRISPR-centric history. So in 2008, people showed that you could reprogram type 1 CRISPR systems to block phage infection, and also that CRISPR actually targets DNA as its substrate. So that began a lot of the frenzy to try to develop these nucleases, because people had already seen the FOC1 ZFN, but we're already starting to also understand that you could reprogram these things with much shorter sequences, which was really the key innovation of the CRISPR nuclease technology. Around 2010, I put this in here because there was so much hard work that went into developing TALENs for large-scale genome editing, and then they quickly sort of gotten forgotten because of the CRISPR nuclease systems. But 2010, there was they showed that you could repurpose TALENs for nucleases and actually develop some pretty large libraries that I think are still commercially available. 2012 is when the Doudna lab first demonstrated the full reconstitution of the Cas9 system in vitro, and that again gave people now the working parts to go into cells and think about doing a much easier reprogramming system than any of the Talon or zinc finger work had offered to be able to do a high throughput genome editing. In 2013, the first demonstrations were shown in eukaryotes out of the Zhang, Marafini, and Church groups. And then in 2014, there were some CRISPR knockout libraries published out of the Zhang and Lander groups. And this is kind of where we came into the fold of CRISPR. So I started in Ryan's lab around 2012. We had just seen the Doudna Labs publications that had just come online, and I was starting to think actually about how to port that stuff over into the kinds of applications that Ryan wanted to develop in his lab. So we began working on let's see, there it is. We began working on the CRISPR systems in early 2013. By 2014, we had filed applications that were describing early versions of the CREATE technology and how to scale the CRISPR system for precision editing. In 2015 is when we founded the company and began our mission to democratize this for the rest of the public. 2016 is when the publication actually came out to describe all of this for the broader community. And I think the main thing to point out here is that, again, the state of CRISPR when we inherited it was that it was really good. You could reprogram and do one edit at one site pretty well, but the systems just weren't in place to make this a scalable technology in the same way they had demonstrated with some of the DNA shuffling or error-prone PCR systems up here in the non-nuclease domains. So
that's great, Andrew. Just 1 second before you move on. So you highlighted the zinc fingers, the ZFNs, and the TALENs, and you mentioned the biggest inhibitor for those taken off was the length of the DNA that was needed to use them. But what else prevented zinc fingers and TALENs from taking off, do you think?
I think they're still very specific. They're really good for some things. I think the problem was being able to build them rapidly and prototype new ones. So you had to do you had to basically synthesize and piece together repetitive DNA elements to then build full TALEN or zinc finger proteins. And that becomes a fundamental challenge at the level of just taking synthetic DNA and piecing it together, especially for repetitive regions. So there was a lot of good publications describing how to do that. But again, those approaches all suffer from the inability to scale quickly because you have to piece so many parts together to build each new TALEN or zinc finger.
And what about the licensing of those two technologies? How did that impact the spread of them? Of
TALENs and zinc fingers?
Yeah.
I don't know much about that story because I haven't invested there, but
well, specifically where I was leading was that I've heard from many people that that part of the fact that it didn't spread was the restrictive or expensive licensing option. So I think it's related to what you're going to talk about shortly in terms of the madzymes that you're going to come onto later. So we can come back to that later.
I am going to address that, and I would believe that. Yeah, I think there is a desire to control the tools that we've seen in the CRISPR space. So.
Great. Okay, back to you.
All right. So moving forward, so I just kind of wanted to dig in and show you a couple examples of what was published to start to shed light on what these CRISPR systems are doing. So there was some great work out of a group in Europe that up here on the top that really they were working with CRISPR systems that were already native to E. coli. There are many types of these systems, and there's one encoded in the E. coli genome. So they started there, demonstrating that basically they could take different components. Let's see if I can get my mouse back on. So different components of either in this system they had the cascade, which does the RNA processing, or Cas3, which is just the nuclease, and these expressed alone have no activity, but when you have them combined with down here the appropriate strand of guide RNA synthesized, then you get complete blocking of the lambda-phage infection process. So this was some of the first evidence people saw that, hey, these systems, A, can be reprogrammed, and B, that they're potentially targeting something about the nucleic acids. There was still some question as to whether it was an RNA targeting mechanism or a DNA targeting mechanism. And in fact, there are different CRISPR systems that are RNA specific or DNA specific, as we now know. But one of the other big breakthroughs at the time was that the Marfini and Sontheimer did some really nice work to show that they could block plasmid infection here. So this is when they have arrays targeting their plasmid, they get zero colony formation post-transformation here. Whereas if they scramble the plasmid, they get good transformation from that DNA molecule. And they also did some really nice studies to show that a splicing RNA, if they targeted the element that gets spliced out, they should knock out the RNA, and that didn't happen. So they were able to piece together that this was definitely targeting the DNA molecule in the cell. So those were big milestones, and actually there were patent filings from this group out of Northwestern on the bottom here that ended up actually getting abandoned by the tech transfer office there. That kind of opened the door for all of the Berkeley Broad debate that you now see going on.
Great. Andrew, I want to just do a time check. We've got about 30 minutes left, and I want to make sure we've got time at the end for Q&A. So just to make sure you can keep the pace.
Yeah, we'll kind of hop through. Yeah. So
yeah, and I want to get on. Yeah, I want to get on to the create chemistry and the exciting instrument. But also just a reminder, this is live. So if anybody has any questions along the way, please type them into the box, and we'll answer them.
Thanks for that, John. All right. So okay, back on the slides. So again, people have been doing in the background a lot of phylogenetic work to understand what different CRISPR systems were out there. The story, as you know, is that people identified effector molecules that encoded all the properties you needed for RNA processing and DNA cleavage in a single polypeptide. So all the work that had been done in these class 1 systems quickly gave way to people focusing on things they could more easily engineer and repurpose, which led to the class 2 systems of Cas9 and CPF1. This is sort of some of the landmark data reducing the Cas9 system to practice, which happened in 2012. And again, just some of the first data that you could knock a gene out using NHEJ-based repair following a cut, or knock a gene in here with GFP getting knocked into an allele in human cell lines. So again, that's where we started. And we also had some data supporting how you would do precision editing, where people were basically doing co-delivery of a guide RNA and oligo in this format, or a guide RNA and a cloned HDR library in this format. And people were seeing high efficiency at a single locus, which is what was done in both of these studies, this one out of the Marafini lab, this one out of the Shinduri lab. But in both cases, again, you are limited to being able to only really study a couple mutations at a time. And this was called out in both, really in this study from the Shinduri lab, it was highlighted that there was a lot of work that needed to go into scaling this up in this composition. So getting to create, we realized that, I mean, a lot of that came from a poor scalability issue. So in all the co-transformation issues that were the sort of editing dogma, people were using single-stranded DNA repair systems, which scale poorly mainly because of the probability of co-transformation. So if you have a random cell that's received a random guide RNA that's now needs to receive the correct donor repair template, that scales as approximately 1/n, where n is the library size. So for a 1,000-member library, you have a 1 in 1,000 chance of getting a cell receiving the right pair of guide RNA donor. And this is, really the basis for how we thought about developing CREATE. So CREATE was really around understanding that we needed to create a covalently linked HR donor and guide RNA construct to be able to deliver precision editing tools to every single cell in our population and get this to scale for library generation. What was done here is you can see that our constructs encoded basically a guide RNA function and a homology arm function on the same oligo, and we could print these in microarray fashion at hundreds of thousands at a time. So the way that the CREATE technology basically works is we would, for a given library design, print these oligos, clone them in parallel, transform them into cells, and get each cell type to have a different edit, where we have now hundreds or tens to hundreds of thousands of different cell types that we've created in a single transformation. Then we can treat these cells either through screening or selection. At the time, we started with selection as a much better or easier tool to employ for the back end. And then you can track the abundance of each genotype in your population by sequencing the resulting plasmid population. So that's where the trans barcode concept for us came from, where the plasmid was encoding both the HR donor and the edit and telling us all the information we needed to know about which amino acid changes we'd made at the genome scale, which was very exciting because now you're talking about a protein engineering tool that scales the whole genomes. So we then in the early work we set about trying to show that this truly did scale up. So we showed saturation mutagenesis type applications, like what protein engineers would be familiar with, where you can do, here we did over 3,000 mutations in a single transformation, and we challenged the DHFR, or dihydrofolate reductase gene, which is a target for trimethoprim antibiotic resistance. So we mutagenized the protein, exposed it to TMP, and then did the plasmid sequencing on the back end to understand what protein variants were really the key winners in this selective pressure. We, in a couple replicates of this study, we reproduced over 30 years of adaptive evolution data and saw many of the hits that other people had described in the AL, adaptive laboratory evolution literature. We also identified a couple really interesting novel mutations that people had always reported combinatorial sets of mutations that could get you to high TMP resistance. And we saw a couple single amino acid changes that got you, as you can see here, this F153R resistance into the near milligram per mil levels of antibiotics. So this is an extremely potent resistance mutation we found in our first application of the technology to this gene.
Andrew, so just to be clear what's going on there, your traditional protein engineering might take an enzyme and you want to increase its efficiency or yield, and you would maybe do one or two changes or tens of changes by picking an area of the enzyme that you want to change. You'd make all these changes. It would take a long time to do them, and then you'd screen them. And what you did in a single reaction here was do 3,000 changes at a time, create 3,000 different changes, very focused on the particular area that you wanted, and then you were able to screen for efficiency on a much higher throughput than in terms of the edit space than anybody's been able to do before. Is that correct?
Yeah, that's exactly it. So this is kind of falls into the deep scanning mutagenesis theme that people are really trying to go after to understand everything from antibiotic resistance to disease phenotypes that we currently sequence basically in natural populations and try to reconstruct. Here we can forward construct it and understand the full phenotypic landscape in a much more rapid time cycle.
Got it. And we have a question that's come in from Shamila Bankia. And Shamila asks, is this system compatible with CRISPR or just Madzime? And you're going to talk about Madzime in a second, but maybe if you can just take that question.
I mean, I will say that just to back up here, we did this initial work with Cas9. We now, the create technology is portable to a variety of nucleases, and that's something we've leveraged to use it with Madzymes, but yeah, this was all done with Cas9.
Got it. Okay.
So, okay, another case study. So we had been looking at some of the adaptive evolution experimentation that was going on. There's some really elegant studies out of a French group, the lead author Tenéon, that was published in 2012, where they had done a year of continuous culture under 42-degree, so high temperature growth selection with a non-ideal carbon source, and they ran 115 of these selections in parallel. So you can imagine a lot of work in the lab to get this all operating and running for that whole time. And then they full genome sequenced they took one isolate from each of those selection processes and sequenced it to 90x coverage whole genome and identified point mutations that had aggregated across the genome for this particular adaptive evolution. What was interesting is that we decided, well, let's see if we can basically validate their study and also think about other ways to do this where we speed up the evolutionary process by orders of magnitude. So we took that, many of those mutations, so we started with just looking at the point mutations that were in the coding sequences. That was easier to code for us initially. And we also tested all the other library designs we had made in this particular study, but we had 645 of the AL-derived set that went into an experiment, and we used the other ones as sort of our control set. We ran fairly short growth adaptations to avoid any kind of adaptive evolution that wasn't coming from our system. And then we isolated the cells and looked at which plasmids had enriched in the population, and sure enough, as you can see kind of down here, this red distribution, this is a number of cassettes we were actually able to retrieve out of the selection, 405 of the 645, and the vast majority of those were actually enriched compared to the synonymous set shown here in dark black. So this was a nice validation that in fact, we could recover and replicate that data and also speed up the entire process and shrink it down to a much more manageable sequencing run on the back end. All that was collected with part of a MySeq run.
And Andrew, we have a question from Vishal, and you can take it now, you can take it later, but what kind of software tools do you use?
All this design and analysis was actually done with custom Python code. So yeah, so we basically I had to code up a lot of that, and we're now, Inscripta is actually developing a full suite of software that's going to make that available for even non-expert, coders to be able to go in and do all of both the front end and back end design and analysis. So, but at this point, it was all custom Python.
Got it. Okay, and in maybe you can address software when we talk about what's going to be coming down the pipeline when you launch the new machine.
Yeah, I'm going to try to speed through this part and get to that. So, this is just one more example. To not beat a dead horse, I'll just quickly say that we challenged some of these libraries with antibiotics as a selection agent, and we're able to quickly map, active sites. So, this was sort of, a nice validation that even drug binding sites were now at the resolution where we can see which residues are affecting drug binding across the whole genome survey. So this is really the holy grail of where we're trying to go with creating a functional genomic tool here. So, and then we've seen further validation for our approach actually has come from a number of different follow-on studies that have published very similar kinds of technology. There's different brand names for each one, but they're effectively the same. The core concept applied to yeast. There's a few additional features in some of these, but I encourage people to look at these papers and do the cross comparison, and you'll see that this is, I think, evidence that the technology itself is going to gain a lot of traction. This is from labs all over the country. So.
Great. Excellent.
Okay. So again, to sort of re-emphasize, when we started with nucleases, this was kind of the state of the art, where you could go to either a few genomic locations and a small variety of edit types to do gene knockout or gene knock-in for precision type editing. You could do genome-wide knockout. People had done some guide RNA screens where you're doing genome-wide knockout, but you're getting, again, low variety of edit types. And the non-nuclease world had sort of been focused on lots of edit type diversity, but few number of loci, because it's just hard to scale all that build. And where Inscripta really is differentiated is that we allow both the high diversity of edit type and number of genomic locations in ways that haven't been possible before for both genome engineering and genome discovery. So this is where we believe we're most differentiated from other tools that have been out there for decades now. And we believe that by enabling unprecedented scale, superior performance, and greater access, that people are going to be able to take advantage of this to run really sophisticated engineering efforts that they weren't empowered to do before. So,
great, excellent.
Okay, so getting into more of the democratization part of this, one of the things that we sort of realized early on is that it was going to be difficult to get customers interested in working with us if we couldn't provide them all parts of the tool suite they needed to perform the editing. So we set to work doing the kind of bioprospecting that's been done traditionally to find novel enzyme variants for a wide variety of applications, and we did that for this for the CRISPR space, which is where we found MAD7, which we received recognition in the Scientist last year as a top 10 innovation. And really this was about us providing our customers better licensing terms and broader access to being able to do CRISPR-based genome editing within the context of our platform. So we characterized the enzyme. This is some early stuff we put out in some of the white papers you can go download from our website, where we're looking at PAM preference and the ability to edit across a number of different genomic loci. We've also had a lot of different people reach out to us and get involved in helping to develop the enzyme for other applications. So we've seen data come back from Horizon. They we had some partnerships going with them where they took it into mammalian cell lines and tested for, in this case, just gene disruption activity, and they saw really good performance relative to even Cas9 and some of the enzymes that have been worked on for already almost a half a decade. So we were pretty excited that we had found such a high efficiency enzyme and that we could translate into our platform.
Great.
And we get this question a lot. So yeah, there is some identity to the other CPF1s, but it's a very low percentage. This is one of the ways in which we prioritize the way we would do the bioprospecting, was to look for distant homologs or distant orthologs that we could use in our platform.
Great, excellent.
So yeah, here at Inscripta, we've got a lot of really talented engineers and scientists on staff who are helping to really broaden. So I showed you some of the data where we really focused on the edit types where we're diversifying the CDS and doing amino acid swaps. But we're now building the tool out to be able to do ribosome binding site or promoter engineering, terminator engineering, all the different sort of genetic parts that you want, and expanding the sort of edit grammar that can be deployed on our system. This is an map of what we did in designing libraries for a lysine biosynthesis campaign, where the ones in blue are all genome knockouts. The ones in green are all promoter engineered variants, where we did in both of those libraries every gene across the E. coli genome. And then the black are stacked up a little higher here because those are saturation mutagenesis within the coding sequences of these genes. So to prove that we can do all that, we've been doing a lot of development work here at Inscripta to get all of the system components working at high reproducibility and robustness. So here you're seeing genomic reads where we're looking at a promoter insertion going in. This is a triple stop codon library going into the in-frame in a gene, and here's some saturation mutagenesis. All of these kinds of validations we're doing both at single locus and whole genome scale currently to vet the technology and make sure it's working as expected for our customers.
Andrew, we have a question that's come in from Shamila again. How does Madzyme compare against zinc fingers, TALENs, and Cas9?
So I don't think it's worth making much of a comparison to the TALEN or zinc fingers, just because there's we're not re-engineering the protein each time. So these still are RNA-guided nuclease systems for the Madzymes. So it's comparable to the kind of activity we've seen with Cas9 in the past, in terms of both the number of sites we seem to be able to edit and basically how robust the enzyme is and how we reprogram it.
Got it.
Yep. So we've taken the approach of what we did with Create and all of the new feature updates that we've made here at Inscripta, and we've run the sort of directed evolution playbook the way that traditional enzyme engineering companies would, but this time at genome scale and across the whole pathway for a large number of edit types. This is sort of a summation of that work, where this was run by a couple of really talented scientists at Inscripta, Dan Held and Eric Abate, and the data collection was done on Agilent RapidFire to do direct lysine tests, and that was all set up by a gentleman named Michael Clay, who is a great member of our application development group, and these guys basically ran an 18,000 member library screen and identified all sorts of improved variants from their first pass running through this. really validating that the tool we built is now working with the efficiencies that we want to be able to deliver for even the screening customers, where we're delivering a lot of mutations with every round of running the instrument. This whole process, again, was sort of rational in that we looked at the proteins, we looked at the pathways and all the things we wanted to target, and we did genome-wide search. All of this tallied up to 200,000 edits that were designed. Of course, 18,000 were tested, and it turns out you don't need to necessarily saturate your testing to get to really good solutions for next rounds of directed evolution, which we're now doing some of that as part of follow-on proof of concept to show our customers just how to teach them how to use these technologies to get large-scale directed evolution processes up and running. We're also kind of hard at work thinking about the challenges of precision editing. This is a big field. A lot of people are working on it. And in both eukaryotic and bacterial systems, there are the preferred route for repair actually tends to be for eukaryotes, NHEJ, which is non-homologous end joining. This basically often results in deletion, insertion, or religation to form wild-type sequences. These are sort of error-prone repair pathways that we don't really want cells to use. So we think a lot about how to promote better HDR or homology-directed repair, where we want to promote the right players to come in and do the homology-directed repair off of a donor template, which is really how our technology works, is delivering that donor template with the guide and forming those precision edits. In bacteria, same problem, except for that there is no NHEJ, so a lot of them just die. So we've done a lot of work in the background to really optimize these systems so that you don't get large amounts of these setup situations where cells want to escape and do something you don't want them to. So we're refining all of that so our customers don't have to. And with the end goal being able to deliver these precision editing products. So this is sort of how we engineer the overall workflow for our customers. So they'll basically from our device, be able to do push button type of operations in an instrument that I'm not showing here because we'll be actually showing stepping into light with that at SynBioBeta in a couple months. But the device will actually deliver the edited cell population, which can then go into pooled or isolated screening workflows that can be, you can do direct detection or different sequencing applications on the back end. This gives you all the genotype and phenotype information you need to feed the next cycle of directed evolution, which goes back into our design and instrumentation process, sort of not pictured here upstream.
So Andrew, everything that you've described so far in terms of high throughput editing, and you described a $200,000 edit library, is going to be wrapped up inside a sexy looking device that's going to sit on the lab bench. You put in your cells, you programmed it on the computer, what you want to do, you hit a button and just like a bread maker, off it goes. How long would a typical library take to create?
Yeah, so I think, doing this like the old-fashioned way, it's hard to estimate exactly. I don't know if you even can, but because there isn't really an old-fashioned way to do it. The way that we envision this is really like a couple weeks timelines that go through each cycle. I think some of the longest parts will be deciding what you want to engineer and what you want to include in your diversity generation. And we are really wanting to get people's feedback and help them build. We're working with some really great early access partners who have great ideas about what to do with that. So, but a typical run on the instrument itself will probably only take about 30 hours for E. coli, and it will be somewhat dependent on cell type. So the number of cell doublings is part of the workflow, and it'll be a little longer for yeast. So you're looking at end-to-end, a design, build, and test. Build itself being the 30-hour chunk, and then the design and test and analysis being sort of the longer chunks. That all will be assisted by our software, and like I said, this could operate on the sort of weeks time scale to get you through each round of directed evolution very quickly.
Got it. And Edgardo has a good question, which many people may be thinking, which is how do you then decide which of the 200,000 you're going to test?
Well, so we are going to have tiered kit offerings where you can start at 1,000, you can do 10,000. So I think, we're challenging people to think big, which I'm going to go ahead and just leave this as the screen while we're talking here.
Sure.
What would you do with 2,000 edits? I think is a great challenge question for people to think about. But I think, you don't necessarily have to go to that scale to get really interesting biological discovery done. So, we're the E. coli genome is 4,000 roughly genes. So we can do, focused types of, like the promoter libraries I was talking about earlier in the lysine pathway. You can do those sorts of modifications, sort of a genome scale with a relatively small number. We just think that the larger that you go in the edit space, you're kind of leaving no stone unturned. And when you're doing a directed evolution campaign, you really want to make sure you're bringing in all of the diversity, beneficial diversity from across the genome into every round. We think that's what's going to make strain engineering a much more routine science in the future. So.
Got it. And can you address a little bit of the breadth versus depth? What should you be thinking about when you're designing your experiment?
Yeah, I'd say that's somewhat going to be, I think, dependent on what the customer's interests are. I think for a protein engineer, it's probably going to be more the depth question. How deep can you go across one sort of gene? For a sort of a pathway or network modeling lab who might want to actually, take some of their models and test them in an experimental system, you're probably looking more at, sort of the breadth question, and then going through that in combinatorial depth as you build up, combinatorial libraries. So I think again, the answer is going to vary, and we're really interested in partnering with our customers and understanding their problems and helping them, address those questions in the right ways. And we have some really great people on staff to help, guide those discussions. So.
Got it. And this is, really part of the innovation that enables you to do the true multiplexing genome engineering. So I think it's a key part of that. When you're thinking about teaching people how to use this piece of equipment, but also how to think about designing their experiments, what are some of the educational challenges that you're thinking you'll have to come across? Because people are not used to doing 200,000 edits at a time. So how are you going to help educate people?
Yeah, no, it's a great question. I mean, I think, we want to build trust that it is something they can do. And then, I think we are faced with the challenge where, we don't want to be, using the technology and competing with customers. We want to train them how to do this and let them explore their ideas. So, I think that's still an open question, how you get people to sort of buy into that larger scale, operation. I think a lot of it will come from just how economical it's going to be for them to do, to run direct evolution using tools like this, and how good the data, how reproducible and reliable the data is going to be. Because a lot of, adaptive evolution and directed evolution studies really suffer from, you get sort of different answers every time you do it. And I think we're building a really robust system here. We've brought in leadership from the NGS world and the diagnostics world, people who've built really robust, assays for, in other fields that are, they're bringing that expertise to directed evolution now. So that's what Inscripta is going to be able to deliver.
Great. We are almost at time, but we have time for a couple more questions. So if you do have any last questions, please type them into the Q&A box. I wanted to remind people that if you want to see this in action, it's going to be launched at SynBioBeta in October. There's also going to be a workshop at SynBioBeta. So if you want to get hands-on experience seeing how it's going to work, then you can come to the workshop. And if you haven't signed up for SynBioBeta yet, then you can use the discount code Inscripta to receive $200 off of your registration fee, but you've got to do it before noon this Friday. So we have one last question from Vishal, and Vishal asks, are you using design of experiment, DOE, to couple the two cycles? And which two cycles is Vishal referring to, Andrew? Do you know?
I think he may be talking about how to iterate. So design of experiment in the next iteration. I'm not totally sure, but I mean, we have, I sort of alluded to it early in the talk, we have people like Richard Fox on staff who, are interested in building those functionalities even into our designer upfront software. That won't be necessarily part of the first launch, but, as we build a customer base, I think, how you plug in data from each experiment and choose which things to recombine in your next experiment. He's a seasoned veteran in that, and we're going to leverage the expertise we have here to build those capabilities for other people.
Got it. So. Great. So we have a couple of questions from Kevin Clancy that I hadn't noticed. He says, any thoughts on how this might work on chloroplasts or mitochondria? And Kevin also asked, do you have a set of equipment you would recommend for a robust workflow that one would need to use with your instrument?
That's a great, so I think we do sort of have a framework of what equipment pairs nicely with this, just from some of the work we've been doing internally. I'd say that's a more detailed answer. We probably would want to, talk to him directly offline. But as far as delivery to chloroplasts, and I think the hard part there is, really probably around how you localize and ensure, the activity is restricted to the compartments of the cell you want. So a lot of the work we do is mostly nucleus centric, but that's a great question. I've not really heard that one yet. So
great. And Kevin, if you want to follow up, just drop me an email. I can connect you to Andrew. Well, excellent. And just finally, you had talked about the business model for Inscripta isn't that you're going to use this machine to create your own commercial applications. You're a true tools platform company. So you're kind of modeling, I would guess, on a company like Illumina. They're on the reading of DNA. You've got Twist and other companies on the writing of DNA, and Inscripta is going to be the editing or the genome engineering company, as your slogan says. So how did you come to that decision not to go after your own products, but to become a tools company? And why is that important for you and for your customers as well?
Yeah, I think the culture at Inscripta is really that to build a good tool, you need to focus on building the tool. And, I think that, we believe that there's a lot of creativity in the scientific community, and people will pick this up and do things that not even we would think of, and expand it in ways that they have for the NGS platforms, and create new functionalities that will expand what the platform can do. We just see it as our mission, our job to get them the best in-class tools. So, yeah.
Great. And any plans for the future? What do you think will be coming down the line in future product releases?
Well, we talked a little bit about, select mammalian cell lines. So we'll probably, I don't know the timeline yet for when we'll be, revealing more of that to the public, but that's a big push for us. Also, how you do, so we will be doing kits that get at combinatorial editing. Those will be sort of rolled out after first customer ship. So first customer ship will be more of the single edit diversity generation kits to get people integrated and started, and the combinatorial kits will be a follow-on to that that expand the capabilities to more the directed evolution paradigm. So
Great, excellent. And I know you're going to be starting to take orders coming to SynBioBeta. So we encourage as many people to come as possible, see the launch, see what's coming down the pipeline, and you'll get to meet the whole Inscripta team at SynBioBeta in October. Andrew, thank you so much for joining us. Thanks for giving us a peek inside of Inscripta and what's to come. It's a very exciting company, very exciting technology, and in terms of why we are passionate about synthetic biology and making synthetic biology easier to engineer, I think Inscripta is about to come out with something revolutionary that's really going to drive home what we care about and how we can make biology easier to engineer. So thanks for co-founding the company. Thanks for coming on to talk to us about it. And we look forward to seeing you in person in October as well.
Great. Thanks, John. Thanks for having me.
Great. Thank you. Have a good one. Thanks, everybody, for joining us.